classes, in the last chapter, we have studied about biotechnological process. So, how the genetically modified organisms or rDNA is made. Artificially, rDNA is made, and this rDNA is inserted into the plasmid. Then the plasmid is inserted into the host. So, actually, what is the process of biotechnology we have seen in the last chapter? So now we are going to start the next chapter. It is biotechnology and its applications. How this biotechnology, this rDNA, recombinant DNA, how it is used in our daily life. As we know, we have studied about biotechnology. It's a technology which is used in human life, in our day-to-day life. So how it is used, how can we apply, that only we are going to study in the chapter. So already we have discussed this biotechnology. It is used in pharmaceuticals, therapeutics, and it is used to make different medicines like antibiotics, vaccines, and it is used to correct the gene, it is used to correct the genetic disorders. So it is used in different field. It is used in agriculture. So the chapter today we are going to study how it is used, in what way it is used, what is the different application of this biotechnology. Today we are going to study. So first of all, we can understand one of the 3 marks question in the board exam. What are the critical research areas of biotechnology? Critical research areas of biotechnology. Critical research areas of biotechnology. So first one you can understand, providing best catalyst, in the form of organisms or enzymes. Second point you can understand, creating optimal condition condition for acting this catalyst, creating optimal condition for acting this catalyst. And third process we can understand, downstream process. So these are the 3 critical research areas of biotechnology. As we know, we are studying in the chapter application of biotechnology. So first of all, we can understand how this biotechnology is used in agriculture. How it is used in agriculture. Point number 1, how can it is used? It is used agrochemical based agriculture. Second one is organic agriculture. And third one you can say it is resistant. Plants are resistant against the pests and insects. So this is the application of biotechnology in agriculture. As we know, you have studied there are some revolutions like green revolution, blue revolution and white revolution. So by the help of the green revolution, they have increased the food production triple. They have increased the food production, but that also not enough for the increasing population. Triple they increased, but that also not enough for the population increase, the population explosion. So next what they have done, they used some technologies. They started to use some practices, and there are some varieties of plants, hybrid varieties of plants and management practices, and started to use agrochemicals. As we know about the farmers are very poor. They cannot use always this hybridization practices, management practices and chemicals. They cannot use always, because all these are the expensive process. Hybridization is one of the expensive process and chemicals are expensive, so they cannot use always these chemicals. And not only that, you can understand these chemicals, it will affect the plants and animals as well as soil. It will affect. So because of that, what is the remedy for that? That's why scientists, they have introduced new technology, new area that we can understand, genetically modified plants or genetically modified bacteria, virus, fungus. Through that we can increase the food production. So first of all, we can understand one of the technology, one of the tool, one of the technology to produce more yield or more plants by the help of one of the technique that we can understand here, tissue culture. It is called tissue culture. Tissue culture. So from the word itself you can understand what is the tissue culture. Something culture with the tissue. So what you can understand here, first of all we have to extract a tissue from the growing tip of the plant. Extract a tissue. So such extract the part, it is called explant. Ex explant. So explant. Extracted that such a part, it is called explant. And this explant has the ability to make a whole plant. It has the potency, it has the capacity to make a whole plant. That capacity is called totipotency. It is called totipotency. So what you have to understand here, explant is the small tissue part and it has the ability to produce the whole plant, that we can say totipotency. And it has some explanation, some process how to make 100 of plants. That process we can say micropropagation. So what is this micropropagation and what is tissue culture, we can study in the next video. So please listen my next video. Thank you friends.